All right, chapter 10, microbial chemotherapy, or sometimes called antimicrobial therapy. This is where we're dealing with antibiotics, right? So chemotherapy is not specific to cancer. It just means chemical therapy. And so that's what antibiotics are going to be. Now, this is a really phenomenal advance in medicine that took place really in the 1930s. So 100 years ago, right, in the early, I won't be able to say that for long, in the early 1900s, right, basically 1 out of 3 children died of an infectious disease before the age of 5. 1 out of 3 kids under the age of 5 died from an infectious disease, right? That has dramatically changed when these modern drugs, these antibiotics, entered the picture. And so really that started around 1930. It made a huge difference in people surviving in World War II over even what we saw in World War I. World War I, a battlefield injury would often become infected, leading to death. By World War II, in the 1940s, we had antibiotics that could treat a lot of those infections. So when we talk about these antibiotics, this is a picture. Let me get the laser pointer again. This is a picture of a fungal colony. Notice how kind of furry it is. And this chemical, this blue goo that's oozing out of the surface, is an antibiotic. So antibiotics technically are substances produced by the natural metabolic processes of some microorganisms, right? So a lot of fungi make antibiotics, a lot of bacteria make antibiotics. They're basically producing these chemicals to inhibit the growth of other microbes, right? They're fighting with each other for who gets access to the nutrients, and they're doing that chemically. So then what humans decided was like, well, wait a second, could we use this as a therapy, right? And so what we're trying to do is to give a drug, an antibiotic, to an infected person that destroys the pathogen without harming the host cell. And so when you think about these are some of the different microbes, right? So different fungal species here and different bacterial species, and you may recognize some of these names, right? So penicillins are made by molds. We've, of course, since then taken their chemical structures and we can produce these synthetically as well. If you've ever had tetracycline or streptomycin, right, these are made by bacteria. We just stole the idea from the bacteria. And so this table is pointing out what an ideal antibiotic would look like. And there's a lot of things in here, and a lot of them would be ideal but aren't really possible. So this idea of selective toxicity is a big deal. And so selective toxicity refers to having something that's toxic to the microbe, but not to the host cells, right? And so that's definitely something we want to find. We have some antibiotics, but they cause organ damage, and so they're very rarely used. They're only kind of a last resort. We do want a chemical that's microbiocidal. We're not just trying to pause the bacteria, we want to kill them. Okay, they need to be pretty soluble, right? We want to basically get these into the bloodstream, which is mostly water, and circulating throughout the body. We want it to stay in circulation, and so what they're referring to here is that basically the liver in particular, one of the things it does in its role of detoxification is break down antibiotics, right? That's not a normal chemical that should be in our body. And so that's going to be primarily broken down by the liver, maybe a little bit by the kidneys, right? Getting filtered out. We'll talk a lot towards the end of this chapter about antimicrobial resistance, the fact that bacteria are developing the ability to survive some of these antibiotics. And so now in our search for new antibiotics, we're really trying to avoid that. Maybe something that kind of works alongside of our immune system. Maybe we already said active in body tissues, delivered to the site of infection, right? Is it actually able to circulate, get where we need it to be? So for example, if you have an infection in the brain, the blood-brain barrier is going to make that very difficult. I guess I should plug in my computer. Okay, we have computer power again. So I was mentioning the blood-brain barrier. So an infection inside of the brain, you need an antibiotic that can actually cross the blood-brain barrier in order to treat that. Of course, it would be nice if it was reasonably priced and doesn't cause any allergies or leave us exposed to other infections, right? So that's a lot to ask for a chemical. And so really all the antibiotics we have, like none of them are perfect, none of them fill all of these. And so there's still research being done and new antibiotics being developed, though not as quickly as we used to. Okay, so just like we saw with our attempt to control microbes on surfaces and things like that, we have a few different targets that antibiotics are going to work against. And so we are going to run through this idea that we can do things like, again, damage the cell wall. This is where I said these chapters are so similar. Damage the cell wall, damage the cell membrane, damage DNA or RNA, inhibit protein synthesis, and then this is the one that really is a little bit different, and this deals with interfering with some metabolic pathways that microbes have. So we will take a look here. Before I jump into that, do notice we'll talk about what's called the drug spectrum. And so you'll hear some microbes being called wide spectrum or narrow spectrum drugs. And basically what we're looking at here, right? So Isoniazid, this is used on tuberculosis, and notice it's really narrow spectrum. It's something we only basically use against tuberculosis, right? So narrow spectrum, it's only going to be effective on a small range of cell types, okay? Or that target component is only present in certain microbes. And I think I mentioned in the last video, Mycobacterium tuberculosis. The mycobacteria actually have a really unique cell wall. And so this is going to be very narrow spectrum. Things that are a little more, say, medium spectrum, I don't know, streptomycin here, we can use it on a few different things. And then broad spectrum, you'd see like tetracycline, right? That's going to act against a bunch of different microbes. And so that can be really important. So one of the things to keep in mind there is, you know, narrow spectrum is actually pretty nice because it's more targeted. Remember that if you start using broad spectrum antibiotics, you're going to take the good with the bad. And so if you have something available that can really kind of hone in on that particular bacteria, that's beneficial. And that's what we're going to start looking at really. It's what we're doing in lab, this idea of, can you identify the microbe causing disease? If you can't, you're just going to give someone a broad spectrum antibiotic, like, hey, let's kill off whatever's making our patient sick. But if you can actually hone in and identify that microbe, then you can really kind of tailor the treatment towards that. Okay, so the cell wall as a target here. Again, it's peptidoglycan. Here's the picture I wanted last time where we have all this cross-linking. Again, the cell wall is protecting against rupturing, particularly in a hypotonic environment. Okay. It is synthesized. So when after mitosis, when a cell has divided and it needs to grow, it is going to have to synthesize more peptidoglycan. Okay. The classic drug here is penicillin. Okay. Penicillin reacts with some of the enzymes involved in the process of producing peptidoglycan. So it creates these weak spots where the cell wall has been growing. That cell becomes osmotically fragile and then can lyse. But again, this is one of those places where the antibiotic is much more effective on a young growing colony than it is on cells that are already kind of in that stationary or death phase. The other thing to keep in mind with penicillin is that most don't get through the outer membrane of gram-negative bacteria. So penicillins are really not particularly effective on gram-negatives, but can be very effective on gram-positive microorganisms. We've started toying with penicillin because it's been such a good drug for us. There is a broad spectrum antibiotic in the penicillin. The other thing I will say is penicillins. It's a whole class of antibiotics. And so anything really that ends in cillin, methicillin, amoxicillin, those are penicillins. We'll talk more about that. But we do have a broad spectrum penicillin called vancomycin, and that can actually get through gram-negative outer membranes. Okay, the cell membrane could be our target. The group here are called polymixins. The only one that you guys are probably familiar with, this is an active ingredient in Neosporin. Okay, so notice here that we're damaging the cell membrane. That cell is lysing, should be exploding. The hard part is selective toxicity, right? So these polymixins are also going to be damaging our cell membranes. And so that's why you'd see something like Neosporin only being used as a topical antibiotic, right, where it's not killing our cells, but could kill by damaging the cell membranes of bacteria. You can use the cell membrane as a target with some other groups of drugs. You can get some specificity. For example, fungal cell membranes have more of a particular lipid called a sterol. And so if you have a drug that attacks those sterols, that's not going to harm your human host. So generally, as a target, the cell membrane provides poor selective toxicity. It's going to cause a lot of damage to the human cells. They become nephrotoxic, toxic to the nephrons, the kidneys, and also neurotoxic. And so again, not our first choice to use the cell membrane as a target. Okay, we also said we can use nucleic acid synthesis as a target. So again, the synthesis is going to be things like DNA replication or transcription. And so an example of an antibiotic that uses this as a target is Cipro, which is technically ciprofloxacin. Again, a very broad spectrum antibiotic, right? Everything has to do nucleic acid synthesis. And so the thing that you'll find here is that if you can be on a short course of these antibiotics, it shouldn't be a problem for the human host because our cells just aren't undergoing, say, DNA replication near as quickly as microbial organisms need to. So a variety, again, of different kind of targets here. You could block the synthesis of a particular nucleotide, right? An A, a G, or a T, or a C. You might be able to inhibit the replication. You could stop transcription. You can do this by what we call cross-linking the double helix, making it so it can't pull apart and get copied, right? Inhibiting something like helicase that should unzip that. You can provide the cell with what we call a nucleic acid analog, basically a mimic. And so it incorporates something into the DNA structure that's going to make it so it doesn't function properly. Okay, so again, Cipro is a really common drug that uses nucleic acid synthesis as a target. We can also target protein synthesis. And so here we see a bunch of ribosomes, and recall that the ribosome is bringing the messenger RNA through. It should be reading the messenger RNA, and we have our little transfer RNA molecules here that bring the correct amino acid. Right? They have an AA there. So what we see is our bacteria have ribosomes, as do we. But if you think about selective toxicity, this is where we go back to that whole idea that bacteria have a 70S ribosome and eukaryotes have an 80S ribosome. There's slight differences, and if we can utilize those differences, we can get selective toxicity. Otherwise, you're worried about you wouldn't want to damage your own ribosomes, your own protein synthesis. One thing you still can get as a side effect is, remember, your mitochondria actually have their own ribosomes, and those ribosomes are 70S, and so you can cause some damage in the mitochondria. Okay, so you don't have to memorize which particular antibiotic does what, but look at how many different ways we've targeted ribosomes, right? So some antibiotics are going to make it so that that messenger RNA isn't read properly. This chloramphenicol, which maybe you've heard of, we have some antibiotics that make it so those adjacent amino acids can't bond, right? That's the whole point of the ribosome, is to bring those adjacent amino acids together. And so if you can't get them to bond, you can't make the polypeptide. We have some antibiotics that keep the ribosome from assembling. So the ribosome is actually, there's a small subunit and a large subunit that float around. And the initiation of translation is when they attach to each other around the messenger RNA and start reading. And so we have some antibiotics that can prevent that. In this case here, we have a tetracycline. It's making it so transfer RNA can't bring a new amino acid in. You're not going to get a protein that way. And then this final one, what they're doing is the messenger RNA has to get pulled through like a tape, right? So we can keep reading the messenger RNA. And here we see erythromycin preventing the ribosome from moving along the messenger RNA. And so again, we won't get a protein to assemble there. So again, kind of a cool idea, particularly if you can take advantage of the differences between bacterial and eukaryotic ribosomes. Okay, this is the one that's different from the last chapter. This is using metabolic pathways as a target. So a metabolic pathway is where, so if you think about it this way, right? A needs to become B, which will become C, which will become D, right, which becomes E. So each of these reactions, right, A becoming B requires an enzyme, right? So maybe enzyme X, enzyme Y, enzyme Z. So we've developed antibiotics that interfere with this metabolic pathway. So here we have a group, the Sulfa Drugs. They inhibit enzyme A in this pathway. This particular pathway, this is the classic example, that bacteria make their own folic acid or folate. Folic acid and folate are the same thing, right? And folic acid is really important in being able to build DNA properly. So you need, right, here's your purines, your pyrimidines, so adenine, guanine, cytosine, thymine, and also amino acids. So you need folic acid to build DNA. And so this is why folate becomes really important during pregnancy, because you have an embryo that's going to divide and divide and divide and divide, and all that mitosis requires actually a lot of folic acid. Humans don't have this metabolic pathway. You only get folate through your diet. So here's selective toxicity. You can interfere with folic acid metabolism, with the building of folic acid. You're not going to harm your host at all because we don't do this, but your bacteria need to. And so here we have some drugs that can interfere with enzyme A. If A doesn't become B, you never get D, right? Here's another drug interfering with enzyme, what is that, Z. You're still not producing folic acid, okay? And so that's what we're looking at by setting up this disruption in a metabolic pathway. Typically, this is done through something like competitive inhibition. So remember, enzymes have that active site and they only bind very specific substrates. We basically are handing that enzyme what looks like the substrate, but it's really our antibiotic. And so then we don't get the product we need, and we can slow or stop the metabolism of these microbes. Okay, so those are your potential targets, those categories. Again, keeping in mind this idea of selective toxicity. So I don't want to go too far into each of the different families of drugs, right? We have over 250 antibiotics. There's 20 different families that we've identified so far. Lots of different names, right? And of course, there's like the trade name and then the generic names and that sort of thing. So you can do that in pharmacology class, but I do want to make sure you have a general idea of what we're talking about here. So again, we talked a little bit about this class called the penicillins. Okay, again, these are produced through microbial processes. So they're natural, they're not synthetic, though we have started making some that are semi-synthetic. So making little tweaks, little adjustments to do things like help them get to gram negatives, that sort of thing. But what you're looking for here, right, is that -cillin ending. That's going to be pretty indicative of a lot of these. So here they're walking you through these different generations of penicillins. Penicillin G is the original. Penicillin, that actually has to come through injection. So this is not the penicillin that most of us have had. Penicillin V is one that we then altered a little bit to help it actually be absorbed through the GI tract. Okay. And then we get a bunch of different semi-synthetics, right? Things that we're trying to make adjustments so that the penicillin can be more useful to us. Most of the penicillin class antibiotics are really well tolerated, right? There's very few side effects in the host because this is going after the cell wall and we don't have a cell wall. So good selective toxicity, but there are allergies. Now that's different than it being toxic, but it is a pretty common allergy. And we do see some resistance growing to the penicillins. Cephalosporins. So this ends up this is another big family of antibiotics. This makes up about 1/3 of all the antibiotics that we use. Here you're looking for the prefix CEPH or CEF or I don't have one on here, KEF. So anything that's like Ceph or Kef, that's indicating that it's in this cephalosporin. It's a fairly broad spectrum antibiotic resistant to most penicillinases. So what we'll talk about is when we talk about the resistance to antibiotics, some bacteria have actually developed a enzyme that breaks down this ring. This is called the beta-lactam ring. And so one of the resistances is the development of a beta-lactamase, an enzyme that can break down beta-lactam. And so cephalosporins then, we see the first generation actually has that beta-lactam. I guess they actually all have that beta-lactam ring, but it doesn't seem to be broken down by those same enzymes. Tends to have fewer allergic reactions. You see here they're showing you 4 different generations, and basically each of these generations we've figured out ways to make them more effective against gram-negatives than the generation before. Okay, so again, just a couple of the major classes. Your book does go into more detail, and I'm not saying it's not worth taking a look at, trying to get it at least familiar in your brain, but when you hit a pharmacology class in nursing school or wherever you're heading, for sure, you'll get into this. Okay, so I don't have a ton of time to go through these different classes, but I did want to point out, right, antibiotics work on bacteria, right? They don't work on viruses. They don't work on fungal infections, right? Antibiotics work on bacteria. We do have antifungal drugs, right? Remember fungal infections like this athlete's foot, right? That is a eukaryote. Fungus is a eukaryote. So we tend to see less ability to have selective toxicity. We do see more toxicity because this pathogen is more similar to the host. Antiparasitic drugs. So here we're showing Plasmodium coming out of a red blood cell. Plasmodium is the causative organism of malaria. And we have a variety of different drugs to treat, this huge variety of different parasites. You may have heard of chloroquine or hydroxychloroquine, right? That is an antiparasitic drug that specifically fights malaria. So we have a few different drugs. They're trying to target different portions of the different stages of the life cycle there. It is originally what was found was a product called quinine. This actually comes out of the bark of a tree that happens to be common in areas that have malaria. And so people were using that tree bark as a drug, and then we developed commercial pharmaceuticals off that. There are anti-helminthic drugs, right? Deworming drugs. Of course, worms are actually eukaryotic animals, and so again, selective toxicity becomes problematic. Some of these drugs are working to actually paralyze the worms, try to get they call this the scolex, like the way it actually latches on off into your intestines with these little teeth. So we're trying to come up with some way to destroy the scolex so it can't hold on. A lot of these drugs end up leading to pretty bad abdominal cramping. Getting dewormed is not the most pleasant experience, apparently. I personally have never been dewormed. Pretty excited about that. Okay, and then antiviral drugs, right? So we do have class of chemical treatments against viruses. Now, remember, this maybe is going to be the hardest selective toxicity because that virus absolutely relies on the host cell for metabolism. Selective toxicity is almost impossible here. So we have antiviral drugs that work in a few different ways, right? Some of them are going to be working to try to keep the virus from being able to actually dock or infect a cell. Some are actually blocking the replication of the nucleic acids, right? Blocking DNA replication or transcription. This is actually remdesivir, the new COVID-19 drug. That is actually its mode of action. It's actually keeping the viral, in this case RNA, from replicating. And then we also have some antiviral drugs that actually keep, that's not what this one's showing, that keep it from maturing, like keep it from assembling. I guess assembly fits under here. And then some drugs also in this category that keep it from actually like hatching out of that host cell. So we actually do have a few different antiviral drugs. This is probably the newest group out of all of those. But there are things we can do, say Tamiflu, right? Trying to slow down influenza A, or acyclovir for things like herpes. HIV drugs are antiviral drugs. That's actually probably been the biggest reason that this class has expanded the way it has, is looking for ways to control the human immunodeficiency virus. Okay, so we do have antiviral drugs. Okay, so that's like the meat of it. When it comes down to it then, what you're trying to do, right, is find a drug that does kill the microbe, that doesn't harm the host, right? And so it kind of inhabits this sweet spot to do what we're looking for it to do. We do run into situations, right, where these drugs are harmful to the host, where we don't get the selective toxicity that we were looking for. So if you hear a drug that says it's hepatotoxic, that's going to be a drug that's affecting the liver, right? So you might see different liver enzyme levels, say in a blood panel, or the liver becoming fatty liver, right? We get fat deposits in the liver as a result of this damage, or hepatitis, which is inflammation of the liver. And the liver is a super important organ, so this is a tough one. We don't really want to see or be using too many drugs that are hepatotoxic. I mentioned nephrotoxic. Those are going to be damaging the kidneys. Other places, we do have antibiotics that affect, say, the formation of blood. And so they lead to things like anemia, or some antibiotics. This is a pretty common side effect. You'll get photodermatitis. So the skin being more sensitive to sunlight. We've got the tetracyclines, for example, seem to interfere with the formation of teeth and bones, right? It's a staining that takes place. So there are side effects to antibiotics, right? They are not just pure. There is, of course, there are allergies to antibiotics. So we talked about the penicillin allergy. That's the most common one. And keep in mind, right, allergies are a result of immune system. So the first time you get penicillin, you won't know if you're allergic or not, right? That's the sensitization. Any subsequent use of penicillin would reveal if you are in fact allergic, right? If you get a rash or respiratory difficulty, or of course the worst being anaphylaxis. Also keeping in mind that one of the problems we see with antibiotics, one of the side effects, is its effect on our own microbiota, particularly the gut microbiota. So here what they're showing, right, these red dots, this is our potential pathogen. This is how C. diff, Clostridium difficile, works. It's a common microbe. Lots of people have it in their GI tract, but it's basically kind of kept in check by the other healthy, normal gut microbes. So this individual, here we see them taking their antibiotic. It circulates throughout the system, and it kills off all sorts of bacteria, particularly if it's broad spectrum. C. diff is extremely hard to kill. And so that's going to be one of the microbes that survives. It now has the whole intestine. It's a free-for-all, so many yummy things to eat, so much room to colonize. And so that pathogen, right, it becomes basically what we call a super infection. It overgrows and is now going to make that person very sick. And in the case of C. diff, potentially even kill them. Okay, so then the last thing, what we're going to close off here with, is this idea of antibiotic resistance or drug resistance. And I pulled this picture because it's kind of a fun cartoon, right? But what we're talking about here is the evolution of the microbe, right? If an antibiotic kills our Helicobacter pylori, right, he doesn't have any offspring. He's dead, gone. But if we get a mutant version, and we looked already genetically at how horizontal gene transfer works and how these mutations get passed, if he survives, all the offspring are going to have that same mutation that allowed this particular mutant to survive. So it's really an adaptive response, an evolutionary response, where the microbes begin to tolerate a certain amount of a drug that would normally kill it, that would normally cure the infection. Antibiotic resistance is not anything new, right? Even Alexander Fleming discovers penicillin in the 1930s, and by 1940 he is warning people, right, that resistance will develop, and we should be careful on how we use this. So again, this mutation often is through the acquisition of genes, right? We talked about horizontal gene transfer, conjugation, transformation, transduction. We've talked extensively about biofilms and how that kind of colonial living arrangement in bacteria can be really conducive for these kinds of gene transfer. I will say these mutations can also be spontaneous, right? Just a mistake. I mean, that's where all mutations originally start, right? Is with some kind of mistake. Again, mutations are really rare, but bacteria replicate really rapidly. So, and I will say most mutations are bad. So an advantageous mutation is even more rare, but we have huge numbers of bacteria, and it's such a strong selective pressure, right, that if you have that mutation that lets you survive, say penicillin, boom, you get to take over that whole area, and your offspring are going to have that gene. They're going to pass it on to their offspring, maybe to their neighbors, whoever's in a biofilm. And so we do see these genes getting passed around. I do want to be really clear that there are actual known mechanisms to this resistance, right? This isn't just like, oh, they're becoming more resistant, right? We know there's a mutation and exactly how it's influencing bacteria to survive the antibiotic. That's the other thing I will super emphasize. You guys have to have this. You do not become resistant to bacteria, to antibiotics, right? You can't say, oh, I'm resistant to penicillin. We can't use that on me anymore. It's the bacteria that are developing resistance. Does that make sense? So here's what we're looking at. The bacteria that have the mutation give them these different mechanisms of drug resistance. And so one of the big ones is a mutation that lets you make a new protein. If the bacteria makes a new enzyme, and here is that penicillinase or beta-lactamase that I mentioned earlier, right, breaking this beta-lactam ring. You're basically inactivating the drug penicillin. You get to keep surviving and reproducing. So really common to have these new enzymes. We also see bacteria actually changing the permeability of their cell membrane to certain drugs, so they don't uptake those drugs. So notice we had to have a certain receptor to let our little antibiotic in, but a mutation of that receptor, if it changes the shape, the antibiotic doesn't get in, you get to survive and reproduce. We have, this is crazy, one of the mutations is for a brand new pump, okay? And so basically some bacteria have a protein that basically can kick out antibiotics that actually do gain entry. And what's super concerning about these is they're often what are called multi-drug resistant pumps, or MDR. And so this is not resistance to a specific antibiotic, say penicillin. This is multi-drug resistance. Any foreign antibiotic is getting the boot. Right, that's what these pumps are for. Isn't that wild that that's a mutation that happened? We see a decrease in some of the binding sites where these antibiotics work, or their affinity. So here, this example, we were seeing a lot of different ways we could interfere with, say, the ribosome. Basically, the bacteria's ribosome is changing enough that the antibiotic no longer works on it. And then finally, we talked about one of our targets being a metabolic pathway. And so here we had a drug, right, that was preventing B from becoming C, right, our enzyme Y. And so we're not making the folic acid or whatever the product was. And so look what the bacteria have done. And they don't have brains. They're not thinking about this. This is a mutation. And if it's advantageous, it gets passed on. So we have a drug that blocks this pathway. Well, now some bacteria have mutated basically to bypass that. So instead of having enzyme Y, enzyme gamma or whatever, right, is now going to make these products in a way that gets us to the end goal. So it kind of just shifts that metabolic pathway, which I want to say is ingenious, but again, they're not thinking about this. I will say, and we can talk about this a little bit more in class, but think about why we hear more and more about drug resistance. In fact, I know you're watching Hunting the Nightmare Bacteria, which should be very enlightening on all of this. The clinical setting, for example, you have patients who are being exposed to all sorts of different drugs. These patients tend to be weaker. So then in the clinical setting, you end up kind of harboring these drug-resistant bacteria. And so now your weak patient picks up, this new MRSA, this staph infection that's resistant to methicillin. And so then you have to give them a different drug, and their own immune system isn't fighting it off well. And so you're letting the bacteria breed essentially in these patients in these clinical settings. And so we see methicillin-resistant Staphylococcus aureus, vancomycin-resistant Staphylococcus aureus. These are becoming more common. So there's a lot of talk about overuse of antibiotics and those sorts of things. We can talk about this. But I will point out one of probably the biggest place that we're overusing antibiotics is actually in agriculture, right? In feedlots. It's some insane number, like 70% of all antibiotics used in the US are actually used in animals, right? And a lot of this use is actually prophylactic. It's not treating sick animals, it's treating them so they either don't become sick, or even more oddly, they've found where some antibiotics make them put on more weight, right? So it's like the bacteria were stealing some of the energy. Well, let's give them the antibiotic and have them put on weight faster. So feedlots have become a place where antibiotic-resistant bacteria are developing, okay? And these same bacteria can infect humans, right? And so if that's a pathogen, if it ends up in the food supply, we're breeding bacteria to cause infections that we now are really difficult, if not impossible, to treat. And so we see something like 23,000 Americans dying every year from bacterial infections that we can't treat. And so again, I know you're watching the scary movie this week, but really thinking about, are we at the end of this antibiotic era? Like, are we going to, in this arms race with bacteria, are we losing? Are we going to lose? So looking forward to hearing your thoughts about that a little bit more in class. All right, you guys have a great day. We'll see you soon.